All right. Hi, everyone. Welcome. Thank you so much for joining. This is the second webinar affiliated with our 2022 Graduate Research Fellows. We are highlighting the projects that they've been working on for the past 2 years. Second webinar of a month-long weekly series. So thank you for joining. Just a plug that every Tuesday for the month of June, we'll be showcasing some of these projects. We're going to get started. I'm going to provide a little bit of an intro background for those of you who are not familiar with California Sea Grant. We are a federally funded and university affiliated organization that supports applied ocean and coastal science for all of California. And we do this through our research grants, fellowships, and extension programming, as well as communications and outreach. And the Graduate Research Fellowship is only one type of the many fellowship opportunities that we provide. We have research fellowships, policy fellowships, and even like extension-related fellowships. And so you can actually visit our website to learn more information if any of these interest you, or you know someone who'd be interested in fellowship opportunities. I believe we have our a couple of our state policy fellowship and Delta Science Research Fellowships currently open for applications. And I think their deadlines are approaching soon. And I will drop the link in the chat in just a moment. But just to give a little bit background on what our Graduate Research Fellowship is, the program started in 2020, and this was to directly support graduate students who are working on applied research, specifically that met our strategic plan focus areas. And these are creating resilient coastal communities and economies, sustainable fisheries, and healthy coastal ecosystems. And so during the 2-year fellowship, these fellows are directly mentored by a research mentor and a community mentor. And then they develop an outreach plan as part of their project to ensure that their research is useful for the entities and communities that are most invested in this type of work. And so, like I said, throughout the month of June, we are super proud to feature these talks of our 2022 cohort and share all of their great work. So again, I'm going to drop a link, all these links in the chat for you, the rest of the schedule as well. And so today we've got 3 presentations. We'll start with Erin de Leon Sanchez from the University of California, Santa Barbara. Then we'll have Katie Kobayashi from the University of California, Santa Cruz. And then lastly, Hannah Kempf from the University of California, Davis. We'll have them present and then Q&A afterwards, and then we'll be good to go. So I'm going to pass it on over to Erin. You can share your screen. Erin's going to be presenting on the future of a vulnerable fishery. Can red urchins adapt to marine heat waves? Whenever you're ready, Erin, you can take it away.
All right, cool. Can everybody see that okay? All good? Okay, perfect. So, hi, I'm Erin. I am presenting today about my project, which is determining whether or not the red sea urchin can adapt to marine heat waves. Oh. Okay, so about me, my pronouns are she, her. I'm a 4th-year PhD candidate in EEMB at UC Santa Barbara. In the Hoffman Lab. I'm also a 1st-generation Filipina American woman, and I love surfing, diving, and seafood, which was part of the motivation for me pursuing this field. So the main motivation for this project, though, is that marine heat waves, abbreviated from here on out as MHWs, are persistent in our local Santa Barbara region. These 2 figures are from the Santa Barbara Coastal Long-Term Ecological Research Site. On the x-axis, we have years. On the y-axis, we have 3 of our LTER sites. And as you can see here, with the colors showing the either number of marine heat waves on our left or number of marine heat wave days on our right, marine heat waves have been consistent and persistent in our region over the past decade. And so that brings me to one of our beloved kelp forest grazers, Mesostrotus franciscanus, or the red sea urchin. They are they're an important ecosystem engineer in our area, controlling kelp abundances, but they're also a key fishery species. So if you've ever had sushi, you might have had uni or their reproductive parts. They're the primary species harvested for gonads, or again, uni, in California since the 70s. They are fully exploited down here in Southern California, as well as in Northern California since the early 2000s. They're consistently one of California's top fisheries by volume each year, and Santa Barbara itself actually accounts for over 50% of California's landings. Nevertheless, this fishery is vulnerable to events such as marine heat waves and is currently experiencing its lowest landings in history. So it is critical to examine the impacts of anthropogenic and climate-related stressors on a key fishery species and conduct research on the red sea urchin's adaptation to changing conditions because of its vulnerability and because the fishery is suffering so much. So here are my 3 objectives. So 1 is to investigate the role of transgenerational effects as a mechanism for buffering offspring from projected oceans temperatures. 2 is to determine if yearly kelp canopy cover correlates to urchin biomass. And 3 is to assess the interaction of thermal stress from marine heat waves and food availability impacts grazing behavior and gonad quality of adult red urchins. So again, we're going to start with the first one. Transgenerational toxicity, aka TGP, sounds really complicated, and it can be, but it is a potential way for organisms to rapidly cope with environmental changes on shorter ecological timescales. So this is where the parental environment alters offspring phenotype. Here is my experimental design to see whether or not adult exposure to marine heat waves may bolster progeny resilience to marine heat waves. So I have 2 treatments, either a fluctuating marine heat wave acclimation or non-marine heat wave acclimation. And I have about 30 urchins each. And then I acclimate them for around 3 months. And then I spawn them and I cross the marine heat wave parents with the marine heat wave parents, non-marine heat wave with the non-marine heat wave. And then I put the eggs and the sperm, and they grow up into larvae in their respective treatments. So either an elevated temperature, 18 degrees Celsius, or a cool temperature, 13 to 15 Celsius. The metrics that I measured to look at TGP include maternal provisioning. So this is like egg size, body size. I did biochemical assays. So I'm looking at protein quantification and lipid quantification, since those are reflective of the energy that the eggs and embryos have. And I also did LT50 in developmental success trials. So LT50 is the lethal temperature at which the population experiences 50% mortality. And I did that in the heat block pictured to our right. So there's a thermal gradient from 15 to 32 Celsius. And I exposed the embryos for 1 hour, and I did this at the gastrula life stage. So here are some results. We see that there are smaller eggs from females that were acclimated to marine heat waves. On the x-axis in both of these graphs, we have treatment, H being marine heat wave, C being non-marine heat wave. And on the y-axis, we either have diameter on our left or egg area on our right. And there is a significant difference between the 2 egg sizes. And from a previous experiment, from a pilot experiment, I found that there were smaller gastrula stage embryos from females exposed to marine heat waves. So not only were the eggs smaller, but in later life stages, as they develop, we see smaller body sizes as well. And here are my results from our thermal tolerance trials. So I found that developmental temperature has a significant effect on thermal tolerance. However, parental acclimation did not have a significant effect on thermal tolerance. And here we see that the LT50 values for the hot parents with cold children is lower than the LT50, or the temperature at which 50% of the population died of the cold parents with the cold children. So it's pretty interesting finding. Those are pretty interesting findings there. And moving on to objective 2, those to determine if yearly kelp canopy cover correlates to urchin biomass. So the motivation for this, and this is kind of tying into the overview of my project, is the Red Sea Urchin Enhanced Status Report. So I went to the CDFW website and I looked at these management needs and directions, what has to be done for this species and what can be done to better manage the species. And I found this table, and this table actually has everything that they still need to find. So I, at first, was just going to look at how kelp canopy cover correlates with urchin biomass. However, I saw the opportunity to address all of these knowledge gaps. Seasonal gonad yield, yearly and seasonal recruitment, as well as, again, the kelp canopy and urchin abundance. So red urchins are important because, like many other species, the quality of the fishery is related to the reproductive cycle. This is a figure from a previous paper from Sarah Tech. On the x-axis, we have month, and on the y-axis, we have gonadosomatic index, which is the ratio of wet tissue weight of the gonad to the whole body weight. And as you can see here, the reproductive cycle is tracked over the months with the gonad index. So for the methods for this project, I am going out into the kelp forest. I dive twice a month to measure twice to 4 times a month, depending on if a collection is successful. But I measure monthly gonadosomatic index at these 2 sites. Again, they're SPC LTR sites, Mohawk Reef, which is a lush reef, versus Arroyo Camado, which is a barren. So this gives us a unique opportunity to compare the gonad yields between a reef and a barren. And once I collect all of these data, as well as the temperature data from these sites, I will conduct a time series analysis on benthic temperature, kelp abundance, urchin biomass, urchin recruitment, and the GSI data that I have collected. So far, I have collected up to June of this month. And on the x-axis, we have collection date. On the y-axis, we have gonadosomatic index. Again, that's the ratio of the gonad weight to the whole body weight. And we are seeing, well, we think that around here might be when they're spawning, because the gonad mass drops and now gametogenesis, the period in which they grow their gonads, is occurring. And it's not as clear from Arroyo Camado, which is the barren, which is pretty interesting as well. But there are clear differences in the GSI from a kelp forest versus a barren. All right, moving on to objective 3, which is to assess how the interaction of thermal stress from marine heat waves and food availability impacts grazing behavior and gonad quality of adult red sea urchins. So I have an experimental design in which I have an elevated temperature, 18 degrees Celsius, to reflect a marine heat wave, and then a non-marine heat wave condition temperature, 15 Celsius. And then I have different abundances of kelp. This is kind of reflecting what's happening out in the field with barrens versus kelp forests, but we're verifying the results in the lab. And then I'm going to feed them for about 8 weeks. I'm collecting the urchins from the barrens, so we're seeing whether or not they're going to be rebuilding their gonads with marine heat waves exposures. And at each week, I'm going to measure the amount of kelp consumed. And then after 8 weeks, I'm going to measure the gonadosomatic index, so the gonad yield, as well as the quality. So again, it's a fishery, so the quality of the gonads is super important. So I'm going to be doing that. And we actually did a pilot experiment for this last summer. I had the privilege of mentoring a California Alliance for Minority Participation scholar, or a Kempf scholar. Her name's Sophia. We did this as part of her summer research experience. We went through the whole thing, and we went and troubleshot all of the logistics for methodology so that I can figure out how much kelp was needed, how to grade the uni, and so on and so forth. One cool thing that Sophia did is she's an artist and photographer. So she took many images of uni that she found online from different grades, and she created this really cool color card that CDFW actually asked her for, so that they can start using it in standardizing uni grades. And this is also helpful for me for my downstream full-scale feeding trial experiment, in which I assess quality. And she also did GI. And in the interest of time, I'm only going to show this as the results. And this is how well our grading scheme went for the project. So as you can see, she has pictures of the uni that she matched using a photography software to look at whether something was grade A or grade C or grade B. So what's next? This summer, I'm going to be doing an internship with the California Ocean Science Trust. I'm also going to be assisting the NSF Authentic Research Experience for Teachers at LTERs with feeding trials, the scale feeding trial this fall. opportunity to do education and outreach with high school teachers. So we're going to be doing a mini version of that right before I do. I'm going to repeat my experiments because urchins die, and I have to redo that. I finished data collection for my GSI, and kelp canopy cover with urchin biomass objective and then analysis. And I will work with collaborators to update the CDFW Red Sea Urchin Enhanced Status Report. And I'd like to acknowledge the Hoff Lab, my PI, Dr. Gretchen Hoffman, former lab mate, my CFW collaborators and community mentors, Derek Stein and Tony Shao, and Nathan Rosser from the California Commission, as well as my under and the funders, California Grant and the NSF. Thank you.
Thank you, Erin. Now we can move into the Q&A portion. If anyone had questions for Erin, please. Yeah, you're getting a lot of claps. That's awesome. It was a great presentation. If anyone had any questions, please feel free to raise your hand using the raise your hand feature in Zoom. You can even come off mute or just add it into the chat.
Sorry, that was so. We only have 10 minutes to cover like our whole project.
It is a lot. You did
Yeah.
a great job, though. You did a lot.
Thank you.
Yes, we've got one from Jonathan.
Hey, Erin, great
Hey.
talk.
Yeah.
I was just wondering, kind of, with purple urchins being so prevalent, especially in kind of southern California and having a big effect on kelp forests. Is there a big reason, like thermal tolerance, like why the red urchin isn't like playing as big of an effect on that as purple urchin?
Are you asking why purple urchins or red urchins have better thermal tolerance? Is that kind of?
Or I guess like where purple urchins are just so prevalent now, versus
Gotcha.
versus red urchins aren't really. Is there like, do you know of like a main reason why that could be?
Yeah, so urchins, this is like out of the scope of this project, but urchins can like metabolically depress themselves for long periods of time. And so purple urchins, there's like all these barrens, and they're not like a fishery species. They're not like a wild fishery species. A lot of places like the cultured abalone farm or even some places up in Northern California will like collect the purple urchins and fatten them up and sell them, but there's not as big of an effort around that. And so that's why I think they're still persisting. I mean, disease outbreaks and warming events do like cause mass mortality events of the purple urchin, but they're still super prevalent. I'm not completely sure why, but there's a lot of factors that go into that. And there's also the loss of predators, there's no sea otters or, yeah.
Cool, thank you.
Great. Last call for any questions for Erin. And if not, that's totally fine. Erin will be on if you think of anything and you'd like to chat her. And speaker Erin and all the speakers today, feel free to put in your contact information if you're willing to, if anyone would like to follow up with you, up to your discretion. But all right, thank you so much, Erin. We can go ahead and move on to our next speaker, Katie Kobayashi, from the University of California, Santa Cruz. We'll be presenting on do wildfires place imperiled salmonid populations at risk of metabolic meltdown. So I will allow Katie to come on and share their screen.
Just working on sharing my screen here. Are you able to hear me in the meantime?
Yes, we hear you. I'm trying to find you so I can spotlight you.
Uh,
There.
I think it's just having me wait while someone else is sharing.
Um,
Tanya, I think you shared with Nancy Kobayashi by accident. Or Nancy is sharing.
okay. Oh, there we go. Are you able to share now?
Yes.
There you go.
How's that look?
Perfect. Thank you. Technical difficulties, but we're on our way. I'll pass it on to you, Katie. Take it away.
Great, thank you so much. And thank you everybody for being here today. My name is Katie Kobayashi, and I'm really excited to get to talk about some of the work that I've been doing for California Sea Grant lately. And I'd like to begin by sharing just a bit about the journey that led me here. And to break the ice a bit, I thought I'd start with a throwback to some of my earliest documentation of my fascination with salmon and trout. They say early exposure to the outdoors promotes career retention in the natural sciences later on. So I certainly serve as a case study on the topic. Fast forwarding a couple decades, and I've had a number of really incredible mentors that really catalyzed my interest in fisheries biology, particularly as an undergrad at UC Berkeley, and then a technician with NOAA's Southwest Fisheries Science Center. And all that led to my current position as a doctoral candidate in the EEB department at UC Santa Cruz. Now, there was a bit of a plot twist along this journey, which I'll mention, and took place while I was right in the middle of my field research for my PhD. And that was the CZU lightning complex wildfires, which is what ultimately led to the research I'll be diving into a bit with you all today. But before I get ahead of myself, I'd like to introduce the main characters of this project, and those are the coastal salmonid populations of Central California, which include coho salmon and steelhead trout. These populations have experienced severe declines due to a number of factors ranging from harvest to habitat degradation, among many others. And this has led to protections for both species under the Endangered Species Act. There's also increasing recognition that salmonids are particularly vulnerable to the effects of climate change. And this is in part because both species exhibit anadromous life histories, where they migrate between freshwater and marine environments to complete their life cycle, which means they're subject to population threats in both marine and freshwater environments. They really get the double whammy. My research generally focuses on the early life history stages for these fish, during which coastal freshwater watersheds serve as really important rearing habitat for early juveniles. And at this stage, the potential threats of climate change are already very busy presenting themselves. This includes rising temperatures and prolonged drought, both of which have direct effects on thermal and hydrological habitat. And moreover, combined, these factors have led to escalations in the frequency and intensity of wildfires, which represents a very immediate and concerning manifestation of climate change, especially in California. Wildfire severity has increased nearly fourfold over the last 2 decades, meaning that these already imperiled salmonids are facing yet another major form of disturbance. Now, it's fairly easy to imagine that there are several potential pathways through which the growth, survival, and ultimately resilience of juvenile salmonids is vulnerable to wildfire. And that's because stream ecosystem function is inextricably linked to the surrounding riparian habitat. On one hand, stream ecosystems depend on the surrounding tree canopy for shade and insulation to maintain temperature, as well as for carbon inputs that form the base of many of their food webs. The surrounding landscape also shapes the morphology and hydrology and chemistry of these systems. And because fire can suddenly and severely alter any one of these variables, if not all of them, the list of potential consequences for salmonids can grow exponentially quite quickly. However, relatively little is known about the relative importance of these pathways, which makes it difficult to prioritize conservation and recovery actions. So in an effort to address this, we developed a framework to synthesize some of these concerns, and that centers itself around between the supply and demand dynamics in stream food webs. On one side, we focus on the prediction that when riparian canopy burns, it will decrease shade and insulation for the stream, leading to increased stream temperatures. And that in turn can influence both metabolic demand as well as energy expenditure in salmonids. And most importantly, what this leads to is this potential threat of what we call a metabolic meltdown, where because these are ectotherms, the increasing temperatures are increasing caloric demand, but simultaneously making foraging more costly. So not only do they need more food to maintain their population, they also need easier access to more food. So it's this kind of compounding cycle. On the other hand, there are several habitat factors that can influence food availability for these salmonids. And those include changes in substrate and carbon inputs into streams. And these really have been shown to vary from fire to fire in terms of, as well as temporally in terms of how food availability responds to wildfire. So to explore this framework, we went to Scott Creek, which is a coastal watershed in Santa Cruz County. It supports both coho and steelhead trout, and also serves as the life cycle monitoring station that's operated by NOAA Fisheries, where my community mentor is based. And Scott Creek was at the epicenter of the CZU wildfires in August 2020, and experienced this gradient of burn severity from upstream to downstream across a number of sites that I had already been collecting data at for my previous dissertation chapters. And between the life cycle monitoring station and my previous work, it provided an ideal opportunity to assess this proposed framework at several different levels. Now I'll mention that some of these, several parts of this project are ongoing, so I'll walk you through some of the preliminary results and highlight some of what's next for us. But broadly, our goals are still grouped into these three main objectives. First, to look at physical habitat and directly quantify burn severity across the watershed. Two, to assess changes in metabolic demand and benthic invertebrate abundance and composition. And lastly, to circle back to what this means for salmonid resilience, and in particular, how it influences salmonid density, size structure, and growth potential. So going through some of the physical habitat results that we have, this is what the watershed looked like in 2019 during one of my early PhD field surveys. And you can see here, I'm standing in the exact same part of the stream 2 years later, and there was you can see even just visually that the effects of the wildfire on this habitat were very severe. Here's another picture from another site. Again, same reference points. You can see the logs spanning the channel, but one has blue sky. And that sun penetration into the watershed is one of the main pathways that we're interested in considering for stream temperature. So results for physical habitat, we found a really clear gradient in burn severity with some differences among the two tributaries where we sampled. First off, there was, as you could tell from those photos, a very clear difference in canopy cover. So in all of these figures, blue will represent the before 2019 conditions, whereas orange represents the post-fire conditions, and there was a drastic reduction in canopy cover across all of our sites, except for this lowest down on the main stem site, which was unburned. Second, we found corresponding decreases in both dissolved oxygen and increases in stream temperature across all of our sites. And lastly, we found several pieces of evidence suggesting that sedimentation and lack of sedimentation is filling in pool habitat within our study sites. So on the left, we have a figure showing the distribution of different substrates, which shows a shift towards smaller, fine, and sand substrates over here on the left. And on the right, we have decreased channel depth across all of our sites, particularly at this upper end of all of these distributions was really truncated and filled in by this loosened gravel. So in terms of next steps, my interest moving forward with this project will be looking at this component of food web supply and demand to assess changes in metabolic demand and in caloric availability. And I'll be doing this via field data from respirometry assays that I collected both before and after the fires, as well as benthic macroinvertebrate samples, with the idea being that I can relate these in terms of the energy demanded versus provided, and ultimately identify where this trade-off in supply and demand is affected by burn severity. And to wrap up, I just wanted to leave on a slightly more positive note. And that is that when looking at some of our initial data for salmonid resilience, we actually found some pleasant surprises. First, when we looked at our fish density data, we actually found increases in fish density across all but one of our sites after the fire, which is both surprising and incredible news. And moreover, we actually found that many of these fish were actually bigger than fish at the same size and age from before the fires. So this is indicative that there may be some differences in productivity going on, and generally a great sign for potential resilience in the system. So to conclude, there are several pathways that are possible for linking fire to salmonids. However, they're often still vague. And our focus has been to link some of these processes through this idea of trophic supply and demand, and use it as a framework for assessing the impacts of wildfire. My interest moving forward will be to more specifically quantify these metabolic meltdown thresholds, and understand when demand is actually surpassing supply. Sorry, my computer froze on my last slide. And finally, these signs of resilience in the system provide insights toward future recovery. So that's all I've got. It's a very long list of acknowledgements because this work has been a huge and very collaborative effort among a number of undergrad mentees, funders, and collaborators, including, of course, California Sea Grant. And I look forward to following up with you all with questions.
Great presentation. Thank you, Katie. Did anybody have any questions? Feel free to add them into the chat or raise your hand, or feel free to come off of mute. You've got a lot of clapping hands, and that's great. I'm not seeing any questions, but I had a question. You mentioned it was a really collaborative project, and I know you had to have a community mentor. So I was wondering if you could speak a little bit more about your community mentor, or how working with the community that you were working with for your project, how that went. If you could share a little bit about that.
Yeah, sure. I'm really lucky that my community mentor is a scientist based at NOAA Fisheries, who I've been working for many, many years now, preceding even this Sea Grant work. And so this fellowship was a really awesome natural extension of the collaborations we already had going. I started working for my community mentor as a technician before I even started grad school. And he's been a huge supporter in this entire process of moving forward through grad school. And I imagine someone I'll continue to collaborate with for many years. So it's been amazing. I've really gained a lot of exposure to what it's like to work in the federal sector. And yeah, I love it.
Well, thank you. If no one has any other questions, but feel free to put them in the chat. Katie will also be on till the end. But we can move on to our final presentation for today. We've got Hannah Kempf from the University of California, Davis, who will be sharing with us how to apply Indigenous knowledge and genetic data to evaluate clam garden restoration potential in Northern California. So Hannah, feel free to share your screen and take it away.
Sounds good. Thank you, Tanya. Okay. Let's see. Can y'all see my screen? Maybe Katie, can you give me a thumbs up? You're the only one I can see. Awesome. Thank you so much. Great. Let me just hide myself a little bit more so I can see my screen. Groovy. Okay. Hi, everyone. Thank you so much for being here. I'm looking forward to sharing with you some of the research I did as a Sea Grant Fellow. Not focusing as much on the genetics part of my research today, but more on the clam growth and carbonate chemistry components. So I've titled my talk today, Investigating the Impacts of Acidification on Juvenile Pacific Little Neck Clams. Can shell hash promote their growth by altering pore water chemistry? So just a little bit of context about me. My name is Hannah. I was born and raised in the Midwest. I'm from Indianapolis, and then did my bachelor's and my master's degree as a combined program in geology at Miami University, which I finished in 2018. In 2019, I moved out to California to pursue my PhD in Earth and Planetary Sciences from UC Davis, where I studied the impacts of ocean acidification on native clams. I am currently a California Sea Grant State Fellow working at the San Francisco Estuary Partnership. And before I dive into the research, I just want to start off by thanking my collaborators, including my research and my community mentors, as well as my many thought partners who have been involved in this research, especially my primary advisors, Sandy and Carlson and David Gold, as well as my collaborators at the Bodega Marine Lab, including Tessa Hill and Alyssa Griffin, Joe Newman and Sim Schneider, both community mentors who were really critical for this project, as well as a number of students that I got to work with last summer as part of a summer fellowship program at the Bodega Marine Lab. So as all of us in this group are likely aware, anthropogenic climate change threatens marine life really across the globe. And that includes marine bivalves, so things like clams and oysters and mussels. And ocean acidification is one of the many stressors associated with anthropogenic climate change. So as there's more and more CO2 added into the atmosphere, a lot of that gets sucked up by the ocean, and that changes the chemistry of the seawater to make it more acidic. And that's a problem for things that build shells out of calcium carbonate because it makes it more energetically costly to do so, and it can actually cause their shells to start to dissolve. However, we know that under acidic conditions, if we actually add in more calcium carbonate in the form of broken pieces of shell or shell hash, that can actually increase the alkalinity and the pH of the seawater and therefore chemically buffer against ocean acidification. It's important to acknowledge, though, that adding shell hash back into beaches, into clams substrate is not a novel technique. In fact, it's one of many techniques that have been used by various indigenous entities across the northwest coast of North America since time immemorial in their active shellfish management. And if this is a topic that you're interested in learning more about, I highly suggest that you look at least the website for the Clam Garden Network, which is a really incredible network of both native and non-native scientists and practitioners working on this topic. For the purpose of my talk today, I'm going to focus on one primary research question and hypothesis. One is that does ocean acidification affect the growth of Pacific littleneck clams? This is a native clam. And can adding shell hash to their substrate promote their growth under predicted future acidification conditions? And we hypothesized that adding shell hash to clams' substrate would moderate the effects of ocean acidification by changing the chemistry of the seawater, and that this would result in greater calcification or greater growth. So to test this hypothesis, we primarily focused on one species called Leucomus diminia. This is also called the Pacific littleneck clam. This is a native clam species to the northwest coast of North America. It's an infaunal clam of the Veneridae family. Infaunal means that it burrows into the sand. In the case of this species, that's only at about 5 to 10 centimeters, so relatively shallow. This clam has been harvested and eaten for millennia, including in a lot of indigenous managed landscapes. It's abundant to this day in the intertidal zone along the western coast of North America, with a biogeographic range from Alaska all the way down to Baja California. This species builds its shell out of the calcium carbonate polymorph aragonite. That's just the type of mineral. And important for this study, the impacts of ocean acidification on this specific species is pretty understudied. So again, we're using this species to ask the question, does ocean acidification affect its growth? And can adding shell hash promote its growth under predicted future acidification conditions? To test this hypothesis, we followed the following experimental design. First, starting on the left, we wanted to obtain juvenile clams. So that involved going into the field to collect broodstock or adult clams and bringing them back to the lab to spawn. We then took that juvenile cohort of clams and exposed them to experimentally acidified seawater in the lab at the Bodega Marine Lab. And in some of our treatments, we added shell hash to the substrate to see what the effects would be. And we monitored 3 different things. Starting on the top right, we monitored the clams' calcification and their growth. We also monitored the influence that shell hash had on the pore water carbonate chemistry of our different treatments. And lastly, we also monitored a genetic response using RNA sequencing. For the purposes of time, I'm going to focus just on these first two parameters for this talk. Our experimental setup looks something like this. Starting on the bottom left corner, we had a total of 4 different treatments, including a control treatment where our pH was kept at around 7.8. We had a shell hash treatment where the pH was also kept at around 7.8. We had an acidification treatment where the pH was at around 7.4. And lastly, we had an acidified treatment where we added shell hash to the substrate. Each of these treatments we ran in technical duplicate. So we had 2 buckets per treatment, or 8 buckets overall, which you can see on the schematic. For this experiment, we ran it for a total of 90 days. And each bucket, we put in 30 baby clams at the very beginning of the experiment. The buckets also were filled with quartz, medium-grained size sand, except for the ones that had shell hash also added to the treatment buckets. And also we fed the clams once a week with algae. And we were monitoring the pH in these different buckets by bubbling in CO2. So it was hooked up to CO2 regulators and probes and whatnot. For the seawater carbonate chemistry, we measured a number of different things, including the pH, the alkalinity, salinity, temperature, dissolved oxygen, and then used some of those parameters to calculate the aragonite saturation state, which basically just describes the stability of aragonite. So again, that's the mineral that makes up the shell in seawater. And we monitored these in both the overlying water in the buckets as well as the pore water. So that's the water that's stuck between the sand grains. And we wanted to monitor pore waters in addition to overlying waters because clams, if you remember, are infaunal, so they are burrowing into the sand. So they're interacting with those pore waters. To measure growth, we looked at a number of different things. I'll just name a couple here. We calculated the percentage of new growth. So that's basically the percentage of growth that happened during the duration of the experiment. We did this by marking the clams at the very beginning of the experiment with a fluorescent dye called calcine, and then measuring the distance from that mark to the ventral portion of the shell, and then dividing that by the overall length of the shell. We also measured things like the weight of the shell and also the weight of the tissue in each individual. Now I'll walk you through some box plots showing just our pH results. So on this box plot, you see on the x-axis, we have O, that means the overlying water in the buckets. The P means the pore water in the buckets. And then we have pH, or the acidity of the seawater, on the y-axis. So you can see here, our overlying water was around 7.8 in our control treatment. That's about what we wanted. And then our pore water was significantly more acidic, hanging out at around 7.3 overall. When we compare this to our shell hash experiment, our overlying water is also around 7.8. That's what we expected, but our pore waters are pretty significantly higher than our pore waters in our control treatments, suggesting that shell hash is buffering acidity. In our acidified treatment, the overlying water was around 7.4. That's what we were aiming for. And our pore waters were very acidic at around 7.1. However, there's some spread in the data, which you can see. And then in our acidified treatment where we added shell hash, the overlying waters were comparable to our acidified treatment on its own, but the pore waters were interestingly actually less acidic than the overlying waters at around 7.45-ish on average. When we used a two-way ANOVA to look at this statistically, we found that the pore water pH varied significantly due to both the overlying pH in the buckets as well as the presence or absence of shell hash. And we found that the interaction between these was statistically significant. I won't walk through the new growth quite as slowly for the purposes of time, but when we measured the percentage of growth on the clams, long story short is that we had really a lot of variability in our data. So you can see generally the data is relatively comparable to one another. I will just note that on average, our acidified treatment with shell hash added was on average a little bit higher than our acidified treatment alone. And when we used statistics to look at this, using again a two-way ANOVA, we found that the differences were statistically significant due to overlying pH, so whether they were exposed to acidification or not, but not necessarily the presence and absence of shell hash. When we used a Tukey test to look at pairwise differences between different treatment groups, we did find that there was a statistical significance between the acidified treatment with shell hash and the acidified treatment. Just very briefly to cover our growth and our carbonate chemistry parameters, we found that shell hash did buffer acidification of pore waters, and this was statistically significant. Also, growth was lowest on average in our acidic conditions, and clams grown in acidified water typically grew more when there was shell hash present. I'll also just very briefly mention that we did a whole another chapter's worth of work on the genetics of this experiment using RNA sequencing. This was done to try and understand the genetic response to acidification as well as shell hash. And we hypothesized that shell hash would basically mitigate some of the acidification response in terms of the genetics. And just really briefly, when we looked at the gene expression profiles of clams that were exposed to acidified conditions and had shell hash added, they were actually the most similar when we think about their holistic gene expression to our control conditions. So this was supporting our hypothesis. I'll acknowledge my funders, including California Sea Grant, as well as the Belinsky Fellowship Program at BML in my home department. Also shout out to the Bodega Marine Lab Aquatics Resources Group who helped me figure out how to get these clams to spawn, which was not easy, as well as, again, my mentors' labs. As far as what's next, I'm actually a very, very recent graduate, and I'm now working as a California Sea Grant State Fellow at the San Francisco Estuary Partnership. I'm working primarily for their Wetlands Regional Monitoring Program. And with that, happy to take any questions if we have time.
Thank you so much, Hannah. Great presentation. Claps are being had virtually. Does anybody have any questions for Hannah?
Hi, Hannah. Great presentation. I was curious about how you determined how much shell hash to add to your experimental buckets and if that amount is like a practically scalable amount to use in natural systems.
Yeah, thanks, Grace. That's a great question. I will just say we ended up doing it by weight percent. So 10% of the weight of the substrate was made up of shell hash. So as far as scalability, I think that's relatively practical. You know, it wasn't like huge amounts of shell hash. That decision to go with 10% was somewhat arbitrary. We asked a lot of different people to get their opinions on what would be, you know, most similar to like a clam garden setting or other places where this technique is being used. And that was kind of the median amount that we settled on. But yeah, and it was mixed into the substrate, I should also say. It wasn't just put on top. I think that that's an important distinction because the pore waters are going to be more acidic for a host of different reasons. So it's more likely to actually buffer if it's mixed in to those pore spaces.
Awesome. Thank you.
Yeah, thanks.
We have about 5 minutes left. If anyone else did have any questions for Hannah. And if not, that's totally fine. We can end early. I'm sure no one would be upset about that. I'd like to give another round of applause and thanks to our speakers today. Erin, Hannah, and Katie, thank you so much and congratulations. And your work is really awesome and thank you for sharing with us today. Another plug is that again, the rest of the month of June, we will be highlighting the rest of our graduate research fellows and their projects. So please tune in every Tuesday, 2 p.m. Pacific time. I've dropped the schedule in the chat once more. So we look forward to seeing them all next week. Thank you very much.